everyone. My name is Sang Park, and from now on, I'm going to introduce about one of our research interests. My research topic is one of the most severe problems in desalination, called membrane biofouling, and its modeling and monitoring. This slide shows the brief information about the research. It can be divided into three parts of step. The first step is biofouling experiments and its monitoring. In this step, we make a membrane biofouling phenomena on the surface of a membrane in a lab-scale reverse osmosis system. And during the operation, real-time monitoring using optic device and water quality analysis are conducted simultaneously. In the second step, we deal with the optic device which name is optical coherence tomography to analyze the degree of membrane fouling. This OCT device can provide the information of membrane fouling in real time without any damage on membrane or operation. Also, its post-process technique provides variety forms of information such as geometry data for computational fluid dynamics. Then finally, we can try to make a model by using both experimental data and monitoring data after finishing the experiments. The purpose of our model is to predict the development of membrane fouling and to define the major fouling index with its quantitative influence on biofilm growth. The schematic diagram represents our experimental setup. The remarkable difference with other experiments is the reverse osmosis module and monitoring device. This module made to investigate the membrane using OCT device. The condition of our experiment is to check the influence of organic matter types, representatively the biodegradability of organic matter as a substance of a microorganism. We are going to progress two cases of experiments. The first case deals with high biodegradable organic matter, and the second case uses low or non-biodegradable organic matter. The comparative result is that as time goes by, we can check that a biofilm is formed much better in the first case than non-biodegradable case. With the visual monitoring to verify the influence of organic matter analysis experiment follows the monitoring. In our experiment, size exclusion chromatography, ion chromatography, excitation and emission metrics, dissolved organic carbon, and specific ultraviolet absorbance are measured with the operation. In this slide, I will explain about OCT device and its basic principle and properties with the reason why we select this device in our research. The left side figure is an example of the OCT system, and the other is the principle of the OCT. Light from a source is directed onto a partially reflecting mirror and is built into a reference. The measurement beam reflected from the specimen with different time delays according to its internal microstructure. The light in the reference beam is reflected from a reference mirror at a variable distance which produces a variable time delay. It is combined and detected that the light from the specimen which has multiple echoes and the light from the reference mirror which has a single echo at a known delay. Then, by comparing those two kinds of echoes, the structure of specimen is determined. Its property is that opticity is non-contact, non-invasive techniques for imaging biological tissues. By using the scattering and reflecting properties of a material and distinguish its structure. And it can be used as a quantitative measurement and qualitative assessment. So, this device is able to be used to investigate the surface of membrane without any damage delay in real time. Moreover, OCT provides the post-image processing. We use two kinds of image processing called 3D modification and quantification. The fundamental OCT image is 2D and cross-sectional image. However, by using image processing, a series of 2D images are able to be one 3D image. Quantitative quantification method is used to quantify the biovolume of the system. from the original image, extract only fouled area using MATLAB and image processing tool, then the extent of biovolume growth is calculated by counting the pixel of the fouled area. Through this process, finally the quantification of biovolume would be determined. The last process of our research is modeling work. In modeling aspects, we can apply our experimental research into two parts: computational fluid dynamics and fouling estimation. To do CFD modeling, 3D modification and geometric conversion processing are needed. After doing this, the hydrodynamic analysis according to the biofilm growth is able to be conducted using COMSOL multiphysics software. In this module, we can simulate the influence of fouling on the membrane and spacer to velocity and pressure variation in the module. To develop the fouling prediction model, we are going to consider about machine learning method with variety of experimental results as input data. In this model, fouling index, which is the dominant factor of membrane fouling, would be used as a key parameter to predict the fouling phenomena during the operation. In conclusion, I will show one of the research figures of a high biodegradable case experiment. This figure is about the research of a high biodegradable water sample during 20 days. As you can see, according to the time variation, the biofilm has grown. Also, this figure verifies the probability of OCT as a tool of membrane fouling monitoring. This is the end of my research presentation, and thank you for listening.